welcoming everybody to the ibiTea today. So my name is Louise, and the overall topic of today's webinar is next generation 3D bioprinting for disease modeling and personalized medicine. We have two exciting speakers with us today from Italy. So our first speaker is Cristina Scielzo. She is the group leader of the malignant B-cell biology and 3D modeling unit at the IRCCS Hospital, San Raffaele, Milan. And she will present a dynamic 3D bioprinted in vitro tumor model system to study the interaction of leukemia cells with their microenvironment. Then afterwards, we have our second speaker. So he is Gianluca Sidonio, and he will afterwards present a microfluidic-based 3D bioprinting method for creating these next-generation in vitro tissue models. And he is an assistant professor and group leader of the 3D bioprinting lab at the Department of Mechanical and Aerospace Engineering at the University of Rome La Sapienza. And we thank you both very much for joining us today and taking the time to enlighten us in this exciting topic. So each of the talks will be approximately 20 minutes, and then it will follow with a 10 minutes Q&A section. This also means that you're more than welcome to include questions as we go into the Q&A section, and then we will do our best to cover all of them. So I would say, let's get started. I hope you have your tea or coffees ready. So Cristina, feel free to share your screen. And I will see, it looks perfect. Good. Then I will hand over the attention to you.
Thank you. Thank you very much. Can you hear me?
We hear you perfectly.
Okay, great. So thank you very much for the introduction. It's a great pleasure for me to be here today and to discuss a little bit about our work that is mainly focused on hematological cancer. But what I'm going to do is to give you some tips that you can, I think, use in general for every disease modeling that you are planning to do in the future. So not only 3D bioprinting, but also 3D modeling more in general. So let me give you a little bit of introduction on what we are doing in order to explain you also a little bit better why we ended up in working on 3D modeling. So we are working on chronic lymphocytic leukemia, that is the most common adult leukemia in the Western country. And what is interesting about this disease is that it's a very dynamic disease. So leukemia cells traffic and home between the blood, the bone marrow, and the secondary lymphoid tissue, where they establish really a complex crosstalk with a supportive microenvironment. And now, actually, I have to say that during the last year, really patient management changed a lot, and it's going very well compared to the past. In particular, BTK inhibitors that act by mobilizing leukemia cells from the tissue into the peripheral blood, where they eventually undergo apoptosis because they lose the protective effect exerted by the microenvironment, or BCL2 inhibitor that directly act by targeting leukemia cells inducing apoptosis, they really work fine. But in any case, patients at the end of the treatment at a certain point relapse and they develop resistance. So nowadays, new immunotherapeutic strategies and combination therapies are under investigation, yet not very easy to test in the models that we have currently in the lab. But so far, CLL remains an incurable disease. So I think that at this point, everybody's asking why this, because a lot of research is going on. And I think that probably we are missing something. We are missing something on maybe on how we are studying leukemia cells. So what we do usually is to take leukemia cells from the peripheral blood. So from this point of view, we are quite lucky because we have a lot amount of primary cells from different patients. So we could envision to do a lot of personalized drug testing too. But still, this has come from the peripheral blood, where we all know that what's really happening that is important for the disease, where the disease actually are proliferating, expanding, developing, and also maybe lacking from therapy is happening into the tissue. And this is the point. So we don't have access to this tissue usually because they are very rare and they actually usually go to the pathologist. So we really need at this point, in my opinion, but also in the general opinion, to generate new humanized model. So also achieving what we cannot achieve in the mouse model so far, to recapitulate physiological microenvironment interaction and dissemination in the tissue, to perform also more relevant functional studies in vitro, to test current but also new therapeutic strategies in the future, in particular immunotherapeutic strategies. So, but what we need to recapitulate? So in our case, we need to recapitulate the complexity of the lymphoid environment, but this is depending on what is the disease model you are working on, you really have to consider which is the tissue of interest and also the multiple cellular components that are part of the tissue. So in our case, mainly the bone marrow and the lymph node, but probably in your case, other tissue with the relevant cell types that contribute to the cancer. But on the other hand, you also have to consider the non-cellular components. Overall, if you are moving towards 3D modeling, such as the stiffness of the extracellular matrix of the matrix where the cells are embedded, the microarchitecture of the tissue, the compressive stress that are present in the tissue and where cells are exposed to. So all these components you really need to take into consideration. So what can we do so far to model the microenvironment and all these components in vitro? So not that much regarding 2D culture. So for 2D culture, you can envision to perform some co-culture experiments um, by using maximum 2 cell types, not much more. You can maybe achieve more physiological models by going by moving to 3D culture. So in particular, 3D culture can be divided between scaffold-free technologies and scaffold-based technologies. Between the scaffold-free technologies, you can find spheroids and organoids. In this case, again, there is not much of the microenvironment, are more cells that are aggregating in a 3D structure, so it's okay from geometry point of view, but still difficult thinking to add a lot of microenvironment here. So you can maybe go more for a scaffold-based technology using scaffolds that can be generated by cell seeding of cells or by cell inclusion of cells, for instance, by using 3D bioprinting. There are also a lot of opportunity by using microtissue, generating so 3D models for microtissues, but here, of course, you need to have access to the tissue samples, tumor tissue, or in general, whatever you need for your purpose. And then you can increase even more the complexity by adding a dynamic culture to your system by using bioreactors or microfluidic devices. So in particular, during my presentation, I will focus on scaffold-based technologies and the use of bioreactors. So, but why 3D model could really be an advantage? I think everybody knows very well this slide. We know that by culturing cells in 2D, you really lack the opportunity somehow to change the stiffness of the substrates, and we do know that the stiffness is important. It's not possible to really reproduce cell-cell interaction in all the 3D dimension, while it's possible in 3D. And also, of course, gradients of soluble factors or drug can be very well represented in 3D, but not that much in 2D culture. But the point here is not really which are the advantages of the 3D culture that are somehow intuitive. I think that the point that we need to make is why we are still stacking with 2D culture. I'm the first still stuck in there for many aspects. The reason, in my opinion, is that 2D culture are cheaper compared to 3D culture, that of course are not that much expensive as mouse model, for instance, but the costs are really difficult to anticipate so far. And also importantly, you know, the 2D culture are very much standardized and are really easy also to scale up in your studies, while it's still 3D culture need to be further standardized. And last but not least, 2D culture are very easy to be interpreted, while 2D culture are 3D culture are not that easy, and not everybody knows how to interpret the results that come from 3D cultures. So, but I really think that is the right moment to bridge the gap and to pass and start using 3D culture. First of all, because FDA officially recognize that we can also do non-animal research in the lab for drug testing, for instance, and by using, for instance, 3D culture, microfluidic chip, or tissue models, and so on so far. So this is an important step overall if you perform drug testing and precision medicine, such as in our case. There is now a high awareness of the limitations of traditional preclinical tool, because we all know that a lot of clinical trials in humans failed during the last year. Now, the feasibility improved in terms of reproducibility of the 3D culture. And also now we know that we can really have an increase of knowledge by modeling the disease by using this technology. So I really think that now we are all convinced that it's time to explore this kind of approach in our laboratories. So what about 3D? 3D culture for CLL, not much is known, and in general, not much is known for hematological cancer, also because at the beginning, so the point here, hematological cancer, everybody imagined that you have to focus on the cells in the peripheral blood, but now we all know that it's very much important also to focus on the microenvironment, so in the tissue. So how we decided to approach this strategy in vitro to model CLL, we decided to approach this aspect in this way. So on one side, we want to recapitulate functional interaction in the tissue microenvironment by approaching a scaffold-based 3D culture approach by using cell seeding or cell inclusion, so cell seeding by scaffold and cell inclusion by 3D bioprinting. On the other side, we want to recapitulate the cellular cells dissemination and promoting also 3D tissue organization in our scaffold by using dynamic growth in bioreactor. So in this case, we are using different bioreactor working under microgravity or under millifluidic approach, or also 3D bioprinted homemade bioreactors. So by the combination of the scaffold-based technology and the microgravity bioreactor, we already had some successful results. And in particular, what we do here, we started by mimicking a bone marrow microenvironment, starting from a model that was already established for multiple myeloma. So another hematological cancer that starts from the bone marrow. So we start from that and we optimize it for chronic lymphocytic leukemia. So we use a spongostand scaffold. The scaffold is mainly on a sponge made of collagen. We seed bone marrow stromal cells and we put it up for 24 hours in the bioreactor under microgravity. After 24 hours, we seed leukemia cells, cellular cells in the bioreactor, and then after 3 days, we have our scaffold with the cells inside. But what is interesting here, that we can perform a double analysis. So we can analyze the cells inside the scaffold interacting with the microenvironment, but also the cells that are outside the scaffold, so more in a, let's say, peripheral blood like settings. So what we immediately realized actually is that the cellular cells remain trapped into the scaffold only if there is the microenvironment. So in the absence of the microenvironment, so of bone marrow stromal cells, you cannot manage to keep the leukemia cells into the scaffold. They all stay outside. But then, and also when they are inside the scaffold, you can see here leukemia cells in green and stromal cells in red. They are nicely interacting each other, filling entirely the scaffold. So at this point, I don't have time to show you, but you also demonstrated that the cells that were inside the scaffold behave like primary cellular cells that are in the bone marrow, while the cells that are outside the scaffold behave like cellular cells that are in the peripheral blood. So this was our validation. But we did also another step forward from this point of view. So we were wondering, can we actually use our model to test target therapies? So I told you that they work, in particular BTK inhibitors, such as ibrutinib, that is used as first line in the clinical practice. They're working by mobilizing the cells from the tissue in the peripheral blood. So clearly, it's not possible to test these in conventional 2D culture models. So we decided to test it in our settings, and actually we could, so in this way, analyze the cells that are mobilized after the addition of the drug and also those that are remained into the scaffold. Using this system also allowed us, by testing other target therapies, to reduce the number of animal models for our experiment. We tested and it worked finely. We measured the cells, the leukemia cells that are mobilized from the scaffold here in the surnatum, and we found that they are mobilized, not all at the same level. And you could also see here from the images that the untreated scaffold where cellular cells are in green are still full of cells, while after the addition of ibrutinib, the scaffold is almost empty. So thanks to this model, we could demonstrate that cellular cells can be mobilized, and actually we're also able to predict patient-specific response to BTK inhibitors in vitro. So now we are implementing these models also for the lymph node environment and also to perform drug testing with other target therapies. But still, we are stuck in this case, to cell seeding. So we need to seed the cells on the scaffold. And this has a lot of limitation. Overall, if you want to improve the complexity of the system by adding different cell types to mimic other tissue. So that's why we decided to move to explore also the 3D bioprinting strategies in order to have a better way to control cell deposition and avoid cell seeding. So let me give just a little bit of introduction and distinguish between printing, 3D printing, and 3D bioprinting. So 3D printing process constructs a 3D object starting from a CAD model that you design at your computer by adding material layer by layer, usually plastic or resin. And thanks to your 3D printing, at the end, you have the rapid production of a customized device that can be a device for your lab, that can be a bioreactor, a lab on chip, or a tool, but in general made of plastic or resin. What is changing when you move to the 3D bioprinting in this case is that you are combining 3D printing with living cells. So they are mixed with smart material that are called bioinks that enable you the generation of customized preclinical model, for instance, as in our case, or tissue-like structure with the defined compartmentalization. So again, you start for the geometry that you wish, you mix the cells with the bioink, that is this really a crucial part, and then you get started with the printing part of the scaffold. You cross-link the scaffold with the calcium chloride, with UV light, or with different strategy to give the rigidity, to have the rigid scaffold that you have at the end that you can put in culture, such as 2D cells grown in 2D. And then the end of the culture, you have really like a tissue on which you can perform all the biological and molecular characterization that you are used to, RNA extraction, histology, flow cytometry, live and dead assay, of course, optimized for the 3D bioprinted scaffold. So that's why when we first started, the real issue was to set up all from scratch, all the protocols. Now you don't have this problem anymore. A lot of protocols have been already established for 3D bioprinting. But the first step that you have to do when you start with this approach is to assess the printability of your cells and all the protocol that you need to use for your experiments. So what we did, we started with the SLS line that was also expressed in GFP, so very easy to follow, to assess printability, the number of cells, viability, the imaging part to see if we can image and follow the cells in vivo, in vitro, and also the distribution of the cells in the scaffold. So once to that, we immediately move to primary cells. And we also, of course, again, we had to find again the best material and to reperform the printability, the number. But now we already learned that by the cell line, so it was easier. So what we immediately found is that the viability is increased in the 3D bioprinted cells primary. In particular, the 3D bioprinted cells were alive up to 28 days. And you have to consider that in 2D, usually after 7 days, they are almost the 30% is dead. So we wanted to see what's really happened here, because we didn't add any microenvironment and nothing at all. Just the fact that they are in 3D in the biomaterial, having a different environment, really promoted this high viability. So we went to perform an RNA-seq analysis comparing 2D versus 3D culture, and we found that actually cellular cells in 3D upregulate genes involved in activation, survival, homing, and adaptation. So they really liked to be in this environment. So we went ahead asking whether we could exploit the 3D bioprinted cellular cells as preclinical model. This is really ongoing. Now we are starting testing different drugs. We started by chemotherapy agents because it's easier, because all the cells respond. And what we see actually is that 3D bioprinted cells show higher resistance to chemotherapy compared to the 2D culture one. So this is important. Also, by increasing the dose a lot, we can see that there is a response also in 3D, but then after 7 days in culture, they become even resistant to the higher dosing. So something is happening here. Probably cells in 3D behave differently compared to the one in 3D culture. This is important to keep in mind and also to start to understand how they respond to drugs in these settings. So, but can we exploit the bioprinting to include the microenvironment, so to incorporate multiple cell types to create a tumor tissue with a defined architecture? So in our case, adding stromal cells from the bone marrow, lymph node, endothelial cells, immune cells, to really build a lymphoid tissue. Yes, you can do that, but please keep in mind, you increase the complexity means that you have to revise all the protocols. So keep in mind that you have to choose the best medium for to culture your cell types. Each cell type has its own medium. You have to redefine the number and ratio of cells. You have to define, most importantly, the most suitable hydrogel. The bioprinted geometry, okay, this is the easiest part. You just have to consider what is feasible with the resolution that you have with your printer, and also considering cells that need to stay viable until the end of the process. And then you have to revise also all your protocols because you have more cell types. Probably it's more difficult to have also a readout compared if you have only one cell type. But the hydrogel part is really important. Please consider taking in mind that. For instance, here, when we start working with UVEC cells, so endothelial cells, they are very, let's say, delicate. So we start with our conventional gel. We had to test many different hydrogel until we find one that was working very well. And then you have to be lucky enough that also the other cell types like this hydrogel. But in case of cells not of hematopoietic origin, you don't have also only to consider that they are alive, but you also have to consider the morphology. So the lymphocytes is round, so easy, but in the case of endothelial cells or in general fibroblasts, you expect to have cells with an elongated morphology. So this is important, and this is your readout to see if it works or not. So then you can move to implement your models to for instance, print cancer cells inside, as in our case, in the stromal compartment outside. And now we are doing that and performing also drug testing in this context. So I think I showed you that 3D co-culture model of bone marrow and also lymph node microenvironment coming soon in the bioreactor allow to elucidate mechanisms involved in leukemia cells retention and mobilization and response to target therapy that can be really envisioned as a tool to test novel inhibitors in hematological cancer, but not only. And that the bioprinting strategy could improve in vitro models by incorporating multiple cell types into a scaffold to create a complex tumor tissue with a predefined architecture that could work as preclinical model for drug testing in a patient-specific way. Please keep in mind also that I think that the impact that 3D culture could have on research is really huge, but there are some points that we have to keep in mind. So the protocol and reagent that you currently are available for conventional 3D culture probably do not apply for 3D culture, so you have to consider that. And future research should focus on the validation of the established model and the standardization overall for the drug testing. I showed you that the response is different in 3D. And comparative studies between 2D and 3D are really needed to establish not that much if 3D is better, but in what it's different. And further research on a common biomaterial is really needed if we want to implement the system for disease modeling and include the microenvironment. So having said that, this is my lab here in San Raffaele that of course I have to thank. They are working hard on this project, and also all our collaborators, and of course all of you for your attention. I'm more than happy to take questions.
Thank you very much, Cristina. That was really exciting. Great introduction to the field and 3D in general, but also some exciting results. We have a few questions, so are you ready if I just fire away? Yeah, okay. So you talk about printability. So what does this cover? That the cells are able to survive printing, and are there certain cells that are not suited for it?
Yes, when I mean printability, I mean that the cells survive after the printing process.
Okay.
So it means that they not only they are not too much stressed, because if you increase the resolution, maybe if you want a fancy scaffold, you have also to, probably you will also decrease the viability of the cells. So you have a little bit to pay
Yeah, take
attention on
into
that. Yeah,
consideration. Yeah,
yeah.
that makes sense. Okay, so when you use many different types of cell for bioprinting, do you mix them first and then print, or do you print one cell at a time and then add one on top of the other, more or less?
So you can have some limitation on that depending how many print head you have. In theory, if you have 6 print head, you can have printing 6 cell types with their own hydrogel. This could be actually the best option because it's difficult to find a common hydrogel. So to mix all together, you need to have an hydrogel that all the cells like. This is difficult. So usually what we do is to compartmentalize. So we print at least in two different positions, so to use different hydrogels.
Yeah, that makes sense to make it, how do you say, viable for the different,
Yeah.
how do you say, preferences. Okay, so once you do 3D printing, do cells stay where they are printed, or are they not moving out of the structures over time?
This is a very good question. It depends from the cells, it depends from the hydrogel, and also depends if you want that they move or not. Indeed, this is another limitation because maybe you have an hydrogel that UVEC cells likes a lot, but leukemia cells not. So what happens? That they don't interact at the end because leukemia cells stay all on their side. They don't go, even though maybe they are attracted from the other cell type, but if they don't like the hydrogel, they don't go.
Mm.
So it's really important to find a common, let's say, environment somehow that could be, you have to balance also what you need.
Do you then compromise, how do you say, the
You have
overall
to
function?
compromise.
Yeah, I guess you would then. Yeah.
Yeah. In some cases, you also want cells to be able to exit from the scaffold. And so if maybe the scaffold is too rigid or the hydrogel is too stiff, it's not going to happen. So you also have to consider the stiffness, the cross-linking part. It's very important to give the correct rigidity, the correct or the rigidity that you want for your experiment.
Sounds like a lot of testing.
Yeah, actually, yes.
Yeah.
I wanted to point it out because important before starting to at least to know that there
Mm.
are so many variability because it's difficult if you start and you don't know
Yeah, that makes
before.
sense. Okay, so what is the size limit of the scaffold to allow cell to survive in your studies?
No size limit because really the hydrogel, oxygen, nutrients, they are really passing very, very good. I think, of course, it's too big. You need maybe too much cells, and it's also more difficult than you probably prefer to have more replicates instead of having a huge scaffold, but it's not really
Can there be a limit the other way around? So can it be too small to not have enough nutrition or so in it, or?
No, not too small. I think that
Okay.
the limitation of the bioprinting, indeed, in terms of dimension, is really
Okay.
that maybe if you want to work with primary cells, so maybe you are not as lucky as me, you have a limited number of cells. 3D bioprinting could be not a good option because you have a minimum limit of the volume that you have to print. Because you have a cartridge, you have to mix the hydrogel with the cells inside the cartridge. So it depends also with the printer that you're using, actually. With extrusion bioprinter, you have to have a minimum volume of hydrogel. So a minimum number of cells. That is
Yeah.
not so small.
Okay. And the next question is actually on your cells. So first there's a compliment, so very interesting. Thank you. And then do you get and print different cell types from same patients? So is it patient-derived cells that you use from the same?
For the microenvironment, no, but this is actually also a choice because you have so little access to the tissue that if you start working with you want to work everything derived from patient autologous, you will have you will be able to perform just 2 or 3 personalized scaffolds. So the idea is to use cells from cell lines or cells that are growing in culture for long, such UVEC cells for endothelium, to generate a model to model the tissue. Of course, is always a compromise, is a model, but anyway, not also a mouse is a model. It works
Yeah,
fine.
that
It
makes
works
sense.
fine.
Yeah, that makes sense. Okay, then there is a question regarding the geometry. So is there like a preferred shape to print in? So is there something you use more than others?
No, definitely. For me, I'm the focus is preclinical models. We don't have. Probably Gianluca will have more, give more importance to the geometry. In our case, really, we are more focusing on having more replicates, not so big, but not also too small, because we want to isolate cells to perform all our readout strategy at the end of the experiment. But not really. The geometry doesn't care
Okay.
for us.
Perfect. There are two more questions to go. So do you think that the available bioprinters are, how do you say, optimized and suitable for bioprinting, or is there still some way to go on them? Like,
Well, I think, of course, there is a lot of implementation overall, as I mentioned, in the possibility to print low volume
mhm.
and also maybe increase the resolution in the bioprinting with the cells, because so far this is can be overall. I say I'm not caring about the geometry also because it's not suitable at the moment to care too much if you want to have live cells, at least with the printer that we have. But I think there is a lot of improvement, but more on the bioprint, I think we really need to improve the materials at the moment.
Mhm. Okay. Do you think dynamic light-based printing can provide better resolution and better printing and ultimately good results overall?
I don't have much of experience on that. I think possibly yes. We are not using this, so I cannot really answer to this question.
Okay. There are actually two more questions coming in. So I think
Vabbè.
they are quite short, so let's just take them before. So is it possible to print into 384 well plates? So I guess very small wells.
So we printed just until 96. I guess that there are printers that can do also that, not
Mhm.
the ones that I'm aware of, the ones that we have in the labs. I don't think so. There are, but depends really from the printer. This is
Yeah, that makes
now
sense.
there are many printers that are coming out in the market, so maybe. But in the 9612 plate, we are printing droplets, and it works very well.
That's already quite amazing, like with
Yeah.
the small geometry. Okay, so last question. Do you slice them up when you analyze them? So I guess your models more or less. Do you then slice them or how do you
Yeah,
analyze
yeah,
it?
yeah,
Yeah.
yeah. So this is really like a tissue. We optimize the protocols to map them, to frost them, and to make slices. So
Mhm.
immunohistochemistry, immunofluorescence. It's like a tissue.
Perfect. I think that was all. Thank you so much again. That
Thanks
was
to
really
you.
exciting.
Thanks
So
to you.
Gianluca, if you're ready, then feel free to share. Then we will take the next talk.
Can you see the presentation?
Yeah, it looks perfect.
Yeah, brilliant. Thank you very much. Thanks, Cristina. Thanks, Luis, for the introduction. Thanks, Cristina, for introducing the 3D bioprinting world to the seminar. So my name is Gianluca. I'm an assistant professor at Sapienza University of Rome. And today I would like to tell you a bit more about our latest advancement on a very niche, very specific way of using 3D bioprinting to create disease modeling systems. So first of all, let me just wrap up on what is 3D bioprinting specifically for my lab. So what we do is to use custom-made printers. So I could see many questions about system that could be developed or better system. We try to make our own. So we have biomedical engineers in the lab that they are trying to engineer new printers routinely. And this is one of the one that we have in the lab, and it's actually allowing us to use a chip that we can attach to the system printing. We can move it around, for example, and here Martina was printing in a gel, in an agarose fluid gel, to create intricate structure that can, support and prevent falling against gravity. So we are able to print cells that they are suspended in this fluid gel system. And following cross-linking, we can actually retrieve the structure and then use it in culture or in implantation or disease modeling, right? So we have the possibility to have multiple inputs and just one output as a channel system. So here we don't have to change print heads. We don't have multiple print heads. We just have multiple images, and we have the ability to pattern really in a stratified manner the final construct. So really to just give a bit of more details, but already Cristina already provided you with lots of them. I just want to make a distinction between what is really biofabrication and what can really do for us. And specifically, biofabrication is really looking into the possibility to use specifically 3D bioprinting for two purposes. Disease modeling and regenerative medicine. So for disease modeling, really is the fact that we can take cells from patients and we can use them to create models to then test drugs, as Cristina was brilliantly explaining. As well, at the same time, we can really use the system, this complex environment, to use the ability of 3D printing to create a new implantable tissue and then can be implanted back into the patient using the patient's own cells, for example, adult stem cells or induced pluripotent stem cells, combine them with inks, then so-called bioinks, to then be used to print the actual tissue, maturated in vitro in the lab, and then to be then readily functionally available and implantable into the needed patient. So let's see. So we really looked a lot at the possibility to regenerate bone. So bone is a highly hierarchical tissue. It remodels a lot during the lifetime, and it's a physiologically relevant tissue. It really structure our body. It can really allow us to walk and stay straight or even be seated right now. As many of you might be, and it typically can get damaged by fractures, and it really is a pain when it's unfortunately getting affected by disease. Imagine thinking about cancer. What we did for many years, we really harnessed the power of materials, really looking, for example, at something called nanoclay. So it's a nanocomposite material made of nanodiscs of 25 to 30 nanometer in diameter. And we use this nanomaterial because it's an incredible system to be combined with a library of materials. And for many publications, for many years, we've demonstrated the fact that we can use this and harness this material to encapsulate cells and print cells, right? So not sitting, but really using this material to have the cells inside, printing them with different 3D bioprinters, and really engineering new bone tissue. So what we did was to demonstrate that we could print lattice structure, that they're really open, an open porosity system, that we can have them be stable for more than 21 days. And think about that this structure have cells inside, okay? So the cells are here inside the fibers. The nanoclay is here. It's here as well inside the fibers. And really, this is what it's allowing the cells to be readily functional. So even after 24 hours, those cells are already triggered to become bone tissue, even without osteogenic conditioning, so in basal media, so without the addition, for example, of beta-glycerol phosphate or any other additives like vitamin D3. This is really like mineralizing over time, up to 21 days, where you can see mineralized nodules. So we tested this interestingly in a CAM model, in a chicken chorioallantoic membrane model, where we had this 3D bioprinter system, implanted them in this vascularized membrane, and looking at the possibility to harness the power of vascularization to drive bone formation. And if you look at this, at the further study that we did, was really looking into the possibility to change the material and to change the function. So be aware that this work here specifically is without cells. And it's pretty interesting because it's even without cells, you have a little bit of mineralization on the control. We know that material, and for those of you who are not familiar with materials, this is alginate, and alginate is coming from algae, and it's well known to be self-mineralizing in vivo, in subcut mice, in subcutaneous implants. You can have some mineralization you see on the low density, but not extensively for 4 weeks. But if you 3D print this material and you combine with different other materials like nanoclay, this laponite, you really find an interesting way of modulating the bone formation. But this was not convincing us, and really what powered us was to use and harness the power of cell work to do the job. So we investigated the system with not only with bulk. I mean, when you read bulk, it's not 3D printed, so it's just a blob of gel. And when we implanted the human bone marrow stromal cells with the bulk gel, so within this bulk gel, you can see there is not much of a difference. So the density scale is still pretty low. But if we 3D print the structure, and this is, bear in mind, this is a lattice structure. This is a grid structure. This is degrading over time and is forming nuclei of mineralization system without the cells. But really what we found, and we were really astonished to find that is when we added the stromal cells or the stem cells component in the bone of the bone, we really found that the tissue was really shining. So we had a large amount of bone formation here, really heterogeneous and really controllable in shape thanks to the 3D printing apparatus. We then wanted to scale up the system, and we wanted to, as Cristina was mentioning, really engineer a new material that was driving the force of the bone formation. So what we did was to actually use human bone tissue to decellularize this tissue, so take out the cell part and the mineral part, blending it with the algae material and the nanoclay and really 3D printing it. And what was really astonishing for us is that this system that we call LAB, so Laponite, Alginate, and bone ECM, it was really driving forward the potential of this system to create new bones. So if you see this portion here in yellow, you can really appreciate the fact that the system is really expressing immediately the alkaline phosphatase. And when implanted into chicken corallantoic membrane model, for example, here you can find that the vessels were very much larger, and the histology was coming out beautifully with this integrated scaffold within the vessels. Again, we did all this with an extrusion-based bioprinter. So basically, with the same system, with similar system that Cristina was mentioning before, but with this system, we found that we were not extremely happy with this because we wanted to engineer a new way of doing things. So basically, we cannot change anything during deposition. We just go faster or make a pressure go higher. We can extrude a bit more, but at the same time, we have no control over this. Distribute spatially the cells by biochemical cues or any other type of assistance to the functionality of the system. And as well, at the same time, we were not able to pattern growth factors of cells. So we were really thinking in what really drives us to think is that we wanted to engineer new functionally graded materials. And if you think about our human body, we are made in gradients. And basically, the interface between different tissues is quite particular. And it could be either on a step base or a continuous base, could be discontinuous or continuous, but we still are made of functionally graded tissues and materials. So really looking at the natural disposition of, for example, of bamboo or bone, these are for bone is a tissue, and this is hierarchically structured, as I was mentioning at the beginning. So to do this, to really harness the power of these in the lab, we are trying to, and with the help with many of the people in the lab, so please let me thank them first. First things first, we're really trying to use microfluidic bioprinting to engineer new systems, either for tissue regeneration or for disease modeling. We are going to focus on some of the disease modeling, but first let me introduce what we're doing right now for tissue regeneration purposes. So first, let me introduce the way that we print. I know this is going to be a bit technical, but I'll try to make it, let's say, as light as possible. In a sense that if you imagine 3D bioprinting is using syringe-based, so extrusion-based is using a syringe, moving around in space and depositing fibers. But these fibers are made of the material within the syringe, and we are not able to control the material that is coming out. We're not able to change the type of fiber which is coming out. But if you have a chip, if you have a microfluidic chip, you have different channels, you have multiple channels of different ways, different shapes. You can change the material, you can change the cell type, and really you end up with a beautiful system where you can have a multiple system of fibers with different architectures, different materials. You can see the hybrid with the Janus fiber with two phases, with two different materials. And at the same time, you can change cells on the go. So this was really drive us to engineer a new system, new microfluidic printhead, thanks to the help of Federico and Martina, two latest PhD students in the lab that now left for good. They managed to create new intricate structure, a new system to help us developing the disease modeling that I will show you in a minute. So what Martina did in a seminal publication was to use this system to create porous structure. So right now, people are just using beakers or system where they can just pour some materials that they then create, like for example for porogen leaching, they can create different type of porosity within the structure, but they cannot really pattern three-dimensional structure, right? So what we did was to really use the power of microfluidic chip attached to a 3D bioprinter to change the porosity. And for example, I want to have your attention here, if it's possible, on this pizza slice. You can see that the density of these dispersions are changing, so they're becoming more dense on the outer side. So we can really, we are able to control the spatial density of these features and really printing different type of material. For example, here we were printing Dexma and nanodroxyapatite, but also we did Gelma, Algin, and some more. We were able to demonstrate the fact that we could print something physiological, for example, like a defect on a jawbone, and really filling it up to then represent, you know, for example, with the highest porosity, the actual trabecular structure of the bone. But again, this is what was not enough. The system was not really biocompatible, so it didn't really get us along with the way that we were doing things. But we wanted to engineer something different, something that could be used in combination specifically with cells. So two brilliant students in the lab, Sajad and Salvatore, they engineer a new open source system, and this is called Moses, and it's out there and it's published, and it's a new type of assembly of 3D bioprinter, which is able to harness the power of microfluidic chips and in combination, for example, with coaxial nozzle. It's able to fit under the hood, and it's able to deposit a library of material. So we did deposit gelatin, gelma in combination with alginate, with laponite, with hyaluronic acid, with chitosan, and really demonstrate the fact this is printable in a number of ways with the high fidelity. In combination with the microfluidic chip, for example, we were able to seamlessly switch between one material and another. So we were able to demonstrate the fact that the switching system was working pretty fine and pretty easily, and actually really fast switching between one and another. And we demonstrate the fact that we could actually construct system that now they're changing ingredients on the z-axis. So you can see the thickness here is changing between one material and another, like this graph here, and it's demonstrating the fact that, for example, you can easily control the way of the interface of the osteochondral defect. So imagine that with this system you can really change the way that you print bone cells, but then spanning into chondrocytes and then really having the matrix building on. Building on this, we recently published just a few weeks ago a solution on the use of this system to print different type of materials. So we wanted to combine 3D bioprinting with high throughput dispensing. So hopefully some of you are familiar with this system, but this is industrially used to dispense materials or drugs. For example, in 96 plate or 384, as we were mentioning before, and really to have the possibility to test in a high throughput manner, for example, drugs. But this was never implemented in combination with 3D bioprinting. So we want to establish a baseline where we were able to print this lattice structure, and they were controllable in size and dimension, and really engineering new materials, including nanoclays inside. But we wanted to scale up the system and to change and use the system to do then high throughput dispensing. So we used 96 watt plate to create a system that was able to dispense not only materials but also drugs. But here, for example, we dispense materials with a different concentration of drugs that was slowly diluting out, and you can see it's beautifully patterning throughout the 96 watt plate, just simply by changing and mixing between one material and another. We're able to partner and independently print. You can see here, for example, different partners, the old IIT logo, and for example, looking at the possibility to print between different type of volumes spanning from 10 microliter and 300 microliter. Now the new system that we have is going even lower than 10 microliter. So we, after doing this, we wanted to combine and harmonize these two systems. So marrying the 3D bioprinting with the dispensing system, we wanted to create a powerful platform. So after printing, the platform is going back, and there is a UV light here, which is actually visible light. Is 405, is cross-linking the system, and we tested the concentration of the drug inside the material. And for example, by including nanoclay, we were able to demonstrate the fact that we can significantly increase the retention of this drug. So after confirming that with the nanoclay power is helping us to retain the drug, we dispense the system in a kind of like in a spherical way. You see in the macro porosity of the printer structure. So we printed in gray, you see here. Hopefully you can appreciate that the yellow system is the compartmentalized drug. So the drug is inside a nanoclay based material, and we are hoping to restrain this compartment in there. So we printed 231 breast cancer cell line into the blue system here in the printing system. And after doing this, we switched the microfluidic chip to dispense the doxorubicin, to localize doxorubicin in the macroporosity. And hopefully you can appreciate that the absence of doxorubicin is really kicking the cell and is making them alive and proliferate quite a lot actually. And when we add doxorubicin and we localize doxorubicin inside, we have a significant decrease of the proliferation, and we have a significant decrease in viability. But if we disperse doxorubicin in the media without localizing it, the cells are doing better actually. They're then proliferating again after the first media change, and nothing is changing much. So we created a system where we can control the modeling of a specific disease and controlling in time and space with the harnessing this powerful system. We wanted to demonstrate also that this is feasible with the CAM system. So we developed an AI tool to then track the vessel, thanks to brilliant postdocs in the lab, Michele, working in Tor Vergata. And we basically found that the localization of the VGF was really doing something, really harnessing the power of and localizing the vasculature around the implanted scaffold. And lastly, my last slide is about the harnessing this system for bone modeling. So, for example, we printed human bone marrow stromal cells from primary cells from patients. We localized the BMP2 around the macroporosity with the microfluidic chip, and we found not much of a difference in viability, but quite substantial and significant difference in proliferation. But what was astonishing was actually the fact that the system with BMP2 was much more functional, as hypothesized, and it was actually readily available for functionalization and actually looking into the possibility to direct more towards the skeletal lineage. And with these, hopefully, I'm finishing in time, and hopefully I give you a quick conclusion on the fact that this is a powerful system. I know it's a bit niche, and it's not as what it's out there about. You might have heard about 3D bioprinting, but hopefully I give you a flavor of the possibility to combine 3D bioprinting with different technology, for example, like microfluidic system, to really scale up and fabricate more functional complex models compared to existing platforms out there. And with these, I'd like to thank the group, specifically the people highlighted, all the collaborators and funding agency, and I'm really happy to take any question if there are any.
Perfect. Thank you very much. What amazing techniques you have developed. So there are already questions coming in. So are you ready?
Yeah.
Then I will just get started. Okay, so the first one, what was the mechanical strength of the bone-based scaffolds that were shown? Whether these are to be utilized in load-bearing sites or not?
Indeed, I think it's up to the point. It's definitely load-bearing sites. It's not going into definitely femur. We are thinking this is possibly going into an arm of somebody or a finger. It's the limitation of the system is indeed the ability to use different type of materials. Specifically, we need to use low viscosity material because of the microfluidic chip inside. But really, what we really believe, and hopefully I showed that the cells are doing the work. So hopefully the stromal cells are putting it out there, the matrix, and creating the new tissue.
Perfect, thank you. The next one was regarding the eye catcher in the beginning, so the Beaker movie. So it's more or less like how to regain the information from there. So how do you regain the information once you seeded your cell or you made your model into this
so this is
gel?
referred to the video? To the initial
I
video?
think it was just like understanding how to regain information for such a beaker, for instance.
Sure. So this is called fluid gel printing. So this is a system, freeform bioprinting. It's out there with many names, but it's the possibility to print into a suspended bath which is allowing us to create complex structure, which are not printable in air, and especially soft structure. We use it to print models that resemble, for example, the brain. And it's quite easy then to cross-link the system after printing, so making the material harden up
Yeah,
to
okay.
then allow for example, for an easy spatula to go in and retrieve it.
Hmm.
There is another system out there. We didn't use it, but it's called Fresh, and it's made of gelatin. And for example, you put that beaker with the gelatin suspended bath into the incubator, that gelatin bath is dissolving, and it's allowing then to regain the system that you printed easily without touching.
Okay, that makes sense. Yeah. Perfect. So it would actually be more or less the same as any other small model. It would just be a matter
Correct.
of transforming the material. Okay.
Correct.
How far are we towards translating clinic? Oh, I think maybe it's more like in a matter of clinics where microfluidics and bioprinting is concerned. So how far are we from translating that towards clinical trials, I think?
Right. So with microfluidic bioprinting, very extremely far. We're very
Hmm.
few people in the world working with that. But with the 3D bioprinting, there are already some cases out there that have been successfully translated. Very poor, unfortunately, very few. There is a large consortium starting now again in Europe to then gain consensus and build the ability of attracting more funding,
Hmm.
specifically to going into clinical trials. So far, unfortunately, not so good, but hopefully in the future, I'm pretty sure we're gonna get there.
Hopefully, yeah. Okay, then there is one last question remaining, at least by now. So maybe a question on the reason for inventing your own printer. So what are the key features that you were looking for, like going
Right.
into the field yourself?
Right. So we do have now about like 7, 8 bioprinters in the lab, all custom made. We did this because we wanted to change whatever we liked. So with unfortunately with the existing system, they're pretty fixed. And as Christina, for example, was mentioning, we there are systems that have multiple print heads. But those systems they incur into delamination. You cannot control cells very well. We're building our own system to be able to control everything we print,
Hmm.
starting from the material, going into the cell positioning, spatial awareness of the cells, where they are, where we can put them, where we can position them, as well as controlling the drug release, or, for example, simply doing something that I showed, like, for example, compartmentalizing the droplets within the pores,
Hmm.
which is something really easy, but with the existing system might be trivial.
Yeah, okay, so you can really adapt it to your needs more precise then.
Indeed, indeed.
Okay, then another question just came in. So how easy is it to adapt a custom-made printer for a non-engineering lab? I guess the option is to use a commercial one rather.
Right. So no non-engineering lab, I guess it's so we there are loads of fun projects out there. We did publish, as I mentioned, the Outward Rex paper on the Moses system. It's very inexpensive. It's below like 700 US dollar, and it's might be trivial for non-engineering,
Mhm.
but I guess there are more simple system out there that they are custom made and that can be assembled, and there are instruction online. I found also online like a DIY system for inkjet bioprinting. There are many, many, many projects, and I've seen biology lab building their own, so I think it's
Yeah, so
feasible.
maybe find a recipe and then follow that a
Indeed,
bit. Yeah,
and apply it. Yeah,
that
yeah, yeah.
that makes sense. Okay, so questions are still coming. I think we have a few more minutes. So, which kind of biomaterials do you think are suitable for print excluding alginate and metasylated materials?
Okay. We have a library in the lab. We make our own. We do use hyaluronic acid. We use methacrylation a lot to allow the possibility to cross-link with light to have
Hmm.
some sort of ability to pick them up and then culturing them up. Um, a lot is now spent on the cellulose material. We have a new library of bone ECM. Collaborators be able to ship cartilage ECM. So really patient-based materials or human-based material is a hot topic. It's unfortunately still pretty uncertain to control.
Hmm.
But definitely, you found the entire myelo that you need for your cell to grow. So it's pretty powerful. So I would say that those are really, really, really cool.
Perfect. Okay. The last one here is regarding these clinical trials. So how custom-made printers are standardized for such trials? So
Oh,
isn't it, more
that's a
more
very good question. I
advanced?
think
Yeah.
that's impossible. So we
Yeah.
need a GMP facility. There is a need of a big industry on the shoulder to be able to translate that. And that's unfortunately the failure of small labs like ours. And it's the powerful potential, I would say. I think the system that we're developing is in a sense is very powerful. But we cannot say that it's much better than the one that are already out there, because the real power lies on the fact that these are is translatable in the end.
Yeah, that makes sense as well. Yeah.
Yeah.
Okay, so the Q&A got a bit quiet now, so hopefully everybody got a chance to get an answer to their questions, and we are perfectly on time as well. So thank you so much for the enlightening talks and all your time and answering these questions. We really enjoyed it, all of us, I'm sure.
Thank you.
And yeah, thanks for it, and hope to see you at another time. So good luck with your further development and research.
Thank you.
Thank you very much.
Very
Good.
much.
See
Thanks.
you.
Bye-bye. Bye-bye.